is Biomed Cafe menu. Ribosome will be presented today. We will talk about ribosome discovery, ribosome structure, ribosome function, and quiz game. I'm Dr. Biomed, cellular and molecular biologist. Hi everybody, welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified and summarized form. Ribosome discovery. They saw something others didn't see. Ribosomes were first observed in the mid-1950s by George Emil Plade using an electron microscope as dense particles or granules. He was initially called Plade granules due to their granular structure. The term ribosome was proposed in 1958. George Emil Plade and two other ones were jointly awarded the Nobel Prize in 1974 for the discovery of the ribosome. Ribosome characteristics. It's a complex cellular mechanism. It's found within all cells. It and associated molecules are known as translational apparatus. It translates genetic code into chains of amino acids. It changes very little between different organisms. It has large and small subunits which come together around a mRNA during translation. Because of non-equal size subunits, is slightly longer in the axis than its diameter. Structure. Ribosome has two subunits, a small subunit and large subunit. A small subunit has two parts, ribosomal RNA or rRNA and ribosomal proteins, RP or R proteins. A small subunit has two functions. It has mainly a decoding function, and it helps to hold the mRNA in place as the ribosome translates it into protein. Large subunit. Large subunit has two parts too, ribosomal RNA or rRNA and ribosomal proteins, RP or R proteins. Large subunit has two functions too. It has mainly a catalytic function and it's bound to the aminoacylated tRNAs. Large subunit has three sites. A or amino acid site binds to tRNA with new amino acids. P or polymerization site polymerizes polypeptide. And E or eject site ejects tRNA without amino acids. Svedberg unit or S. Is the unit of measurement describing the ribosomal subunits and the RNA fragments. Is a measure of how long it takes a molecule to sediment out of solution in the centrifuge. The larger the molecule causes, the larger the S number. Ribosomes types. Ribosomes, according to organism, can be divided into 5 groups. Eukaryotic ribosomes, prokaryotic ribosomes, archaeal ribosomes, plasturibosomes, and methoribosomes. Eukaryotic ribosomes. Eukaryotes have 80S ribosomes. A small subunit is 40S. It has one rRNA, 18S with 1874 nucleotides, and 33 R proteins. Large subunit is 60S. It has 3 rRNA, 28S with 4718 nucleotides, 5.8S with 160 nucleotides, and 5S with 120 nucleotides, and 49 are proteins. They are located in cytosol. Their diameter is between 25 and 30 nanometers, or 250 to 300 angstrom. Prokaryotic ribosomes. Prokaryotes have 70S ribosomes. A small subunit is 30S. It has 16S rRNA with 1542 nucleotides and 21 R proteins. Large subunit is 50S. It has 2 rRNA, 23S with 2904 nucleotides and 5S with 120 nucleotides and 31 R proteins. Prokaryotic ribosomes are smaller as eukaryotes because they contain less proteins and RNA. Their diameter is around 20 nanometers. Archaeal ribosomes. Ribosomes in archaea are the same as prokaryotes. Plastiribosomes. They are ribosomes in chloroplast that are as same as prokaryotic ribosomes. Mitoribosomes. They are ribosomes in mitochondria that are as same as prokaryotic ribosomes. Ribosome types. Ribosomes, according to location, can be divided into two groups: free ribosomes and membrane-bound ribosomes. Free ribosomes. They are free in the cytosol. Free ribosomes can move about anywhere in the cytosol. Proteins produced from free ribosomes can release in the cytosol and use within the cell. Membrane-bound ribosomes. In eukaryotic cells, they are on rough endoplasmic reticulum or RER. When a ribosome begins to synthesize proteins, the ribosome making this protein can become membrane-bound. Proteins produced from membrane-bound ribosomes can use within the plasma membrane or can expel from the cell via exocytosis. Now I want to compare free ribosomes with membrane-bound ribosomes. Distribution of free ribosomes in the cytosol, but membrane-bound ribosomes bind to RER membrane. Free ribosomes have not ER targeting signal sequence, but membrane-bound ribosomes have ER targeting signal sequence. Similarity between free ribosomes and membrane-bound ribosomes. They are identical in structure. Ribosome biogenesis. In bacteria, Ribosomes are located in cytoplasm. Their biogenesis is through the transcription of multiple ribosome gene operons. In eukaryotes, they are located in cytoplasm and nucleolus. Their biogenesis is through the transcription of ribosome genes. Ribosome function. Ribosome plays an important role in producing proteins. Key points of ribosome. The ribosome is a complex cellular machine. It's largely made up of specialized RNA known as ribosomal RNA or rRNA, as well as dozens of distinct proteins. Ribosomes consist of two subunits that fit together and work as one to translate the mRNA into a polypeptide chain during protein synthesis. Thank you for watching. Please subscribe to this channel, and if you like the contents of this video, please like it. Now it's time to the quiz game. I have put the link to the quiz game in the description of this video. Download it for free and enjoy it. Until the next video. Bye!